The Black Death, one of history's most devastating pandemics, ravaged Europe in the 14th century, claiming over 25 million lives. While this catastrophic event may seem like a distant historical footnote, recent scientific research suggests that its impact continues to reverberate through our genes, influencing modern human health in surprising ways. This article explores how the Black Death and other ancient plagues have left an indelible mark on our genetic makeup, and how these evolutionary adaptations present both advantages and challenges for contemporary populations. In 2022, an international team of researchers published a groundbreaking study in the journal Nature, hypothesizing that the Black Death's enormous death toll acted as a massive natural selection event. Their premise was that survivors likely possessed genetic advantages that helped them withstand the plague. To test this theory, the team embarked on an unconventional research method. They obtained DNA samples from several hundred individuals who died before, during, or after the plague, focusing on grave sites in London and Denmark. The researchers' primary goal was to identify specific gene variants that became more prevalent in the surviving population compared to the pre-plague population. They narrowed their focus to approximately 400 immune-related genes out of the human genome's roughly 20,000 genes. This targeted approach allowed them to analyze changes in gene frequency that might indicate protective effects against the plague. Their analysis yielded 4 sections of DNA that likely contributed to survival, with one gene in particular standing out, IRAP2. The researchers identified 2 variants of this gene, with one version expressing significantly more of the gene's product than the other. Further investigation revealed that these variants function differently at the cellular level. The more protective variant, dubbed IRAP2 type A, produced full-length functional proteins. In contrast, the less effective variant, IRAP2 type B, resulted in truncated proteins that were quickly degraded by cells. IRAP2's role in immune cell training, pathogen clearance, and modulation of inflammatory responses made it a prime candidate for enhancing survival during the plague. The researchers estimated that individuals with 2 copies of the IRAP2 type of variant were 40% more likely to survive the Black Death, a significant advantage given the plague's devastating mortality rate. This finding provides compelling evidence of how severe infectious diseases can shape human evolution over relatively short periods. However, the story doesn't end with this apparent genetic triumph. The same IRAP2 variant that offered protection against the plague has also been associated with an increased risk of Crohn's disease, an inflammatory bowel disorder. This paradoxical relationship exemplifies a phenomenon known as antagonistic pleiotropy, where a gene has multiple, often conflicting effects on an organism's fitness. The IRAP2 case is not an isolated incident. Among the 4 variants initially identified as protective against the plague, another has been linked to autoimmune diseases such as lupus and rheumatoid arthritis. These findings suggest that the very genetic adaptations that helped our ancestors survive deadly pandemics may be contributing to the prevalence of autoimmune disorders in modern populations. To further explore this concept, a 2023 study examined 10,000 years of genomic data to assess the extent of antagonistic pleiotropy in human evolution. The researchers discovered a clear trend. As our genes became more adept at protecting against infectious diseases, we simultaneously became more vulnerable to autoimmune conditions. Specific examples from this study include a genetic variant that increases the risk of inflammatory bowel disease, IBD, while decreasing susceptibility to norovirus infections, and another that offers protection against COVID-19 but also raises the risk of IBD. These findings suggest a complex evolutionary trade-off between protection from acute infections and susceptibility to chronic inflammatory conditions. The timing of these genetic shifts is particularly intriguing. Most of these changes appear to have occurred since the beginning of the Bronze Age, approximately 4,500 years ago. This period coincides with significant changes in human society, including urbanization, increased travel, and the domestication of livestock. These developments would have exposed populations to a wider array of pathogens, driving selection for traits that enhanced resistance to infectious diseases. The connection between enhanced immune responses and autoimmune disorders makes biological sense. An immune system primed to react strongly against pathogens, like the plague bacterium, might also be more prone to overreaction against the body's own tissues. This delicate balance between protective immunity and harmful autoimmunity is at the heart of many modern immune-related disorders. The implications of this research extend beyond historical curiosity. Understanding the genetic legacy of ancient plagues can inform our approach to current and future pandemics. It highlights the potential long-term consequences of intense selection pressures on human populations and underscores the complexity of genetic adaptation. Moreover, this research sheds light on the origins of autoimmune diseases, which have become increasingly prevalent in developed countries. The hygiene hypothesis suggests that our modern, sanitized environments may contribute to the rise of these conditions by depriving our immune systems of the microbial exposures they evolved to expect. The genetic evidence of antagonistic pleiotropy adds another layer to this theory, suggesting that our very DNA carries the echoes of our ancestors' struggles against infectious diseases. For individuals of European, Middle Eastern, or North African descent populations most affected by the Black Death, this research may have personal implications. Those dealing with autoimmune diseases might find a surprising connection to their distant ancestors' survival of one of history's worst pandemics. However, it's crucial to note that genetic predisposition is just one factor in the development of autoimmune diseases. Environmental factors, lifestyle choices, and other genetic variables also play significant roles. The complex interplay of these factors underscores the need for personalized approaches to medicine and public health. Looking forward, this research opens up new avenues for understanding and potentially treating both infectious and autoimmune diseases. By unraveling the genetic basis of our immune responses, scientists may be able to develop more targeted therapies that enhance protection against pathogens without exacerbating autoimmune tendencies. In conclusion, the genetic legacy of the Black Death serves as a powerful reminder of the enduring impact of historical events on human biology. It illustrates the intricate balance between evolutionary adaptation and health, showing how traits that once ensured our survival can present new challenges in different environments. As we continue to face global health threats, including new pandemics, understanding our evolutionary history becomes increasingly important. The story written in our genes by the Black Death and other ancient plagues offers valuable insights into human resilience, adaptation, and the ongoing dance between our immune systems and the pathogens that challenge them. This research not only deepens our understanding of the past, but also provides a framework for addressing current and future health challenges. It reminds us that in the grand tapestry of human evolution, every thread, even those woven by tragedy, contributes to the complex pattern of our genetic heritage.